and welcome everyone to 2021, our first speaker for our new year that we have high hopes for. Just for everyone's information, we're starting a new series in the FES Center called Behind the Science. It's going to be more of an informal discussion with people that provide services that want to reach out and do different kind of collaborations. So Curtis Tatsuoka is our first speaker. You probably don't know him. He's a biostatistician that we've recently contracted with to provide support for FES Center investigators. He's in the Case Western Reserve University, and he'll be talking about the kinds of work that he does. And I invite you to take a look and see how it aligns with your needs and your research projects. Secondly, our next speaker is also from Germany, from Berlin this time. I'm probably going to mispronounce his name, Sergio Sokadar. He's a physician in Berlin, and that will be on February 11th. It will be our next neuroprosthesis speaker. And finally, I'm going to introduce our speaker. So our esteemed speaker today is Thomas Stieglitz. He's an old colleague from the broader FES community. We've known each other for a long time, and many of you here today will know him as well. And I already told him, but I'd like to thank him publicly for doing this at 9 p.m. Local time. It's very nice that you're able to join us today. So Thomas is the head of the laboratory IMTech Institute. It's an Institute for Microsystems Technology, and in the Laboratory for Biomedical Microtechnology at the University of Freiburg in Baden-Württemberg in Germany. Thomas has his master's degree. The equivalent, I guess, has a different title, but from the University of Technology in Karlsruhe, and his PhD equivalent from Saarland University, also in Saarbrücken, Germany. Thomas's work is focused on microfabrication and really electrode arrays. If I hope I have that pretty correct. And so he definitely a complementary technology to a lot of the work that we do here in the FES Center. So without further ado, I'll turn this over to Thomas. Thanks again.
Yeah, thank you very much, Robert, for that kind introduction. It's a real honor and pleasure for me to be in your seminar series. And as Robert said, we know each other for such a long time. Honor to me that Thomas Mortimer is listening. Your papers were the first that I read about cuff electrodes and what they do. And before I share the screen, just want to tell you, I assembled slides that way. As Bob said, it's a complementary technology to yours. And I like to show you the efforts and like to convince you that this technology might have some advantages. Of course, it comes with some disadvantages, but probably competitors tell you. I will show you the advantages only. How one can use micromachining to make nerve interfaces. And will share the screen now. And we'll start from the first slide. So my talk, next 45 minutes, will be about flexible microimplants for neuroprosthetics and bioelectronic medicine. So, and what can you expect there? Since I always run out of time at the very end, I decided to show the group and all the funding agencies at the very beginning that I do not forget them. So that was my group, together with those guys who set up 2 companies some years ago. We're now about 25 persons in the group, and things that I present today have been funded through plenty of grants from federal ministries, from our National Science Foundation, DFG, and the European Union. So, but more interesting for you, what can you expect? Today, just briefly, I want to go through some thoughts. I know that in the US, there's plenty of funding for neuroprosthetics over decades, but other parts of the world do not fund that so explicitly. So why could that be good to spend money on that? And then I want to go into some fundamental requirements, reflecting a little bit where we are in the state of the art of implantable technology today. And when having done that, I'll start my wild journey through my last 20 years of research development. So when I started with micromachining, what was the starting point? What challenges do you have to face? And how does it look today? And what is it good for? And I thought I'll show you some brain probes, mainly from the neurosciences, share the technical side of sensory feedback. So Silvestro Michera was some weeks or months ago in this seminar series, so I tried not to steal all his videos, but just show you one of that and discuss a little bit more on the technology side. So how do the nerve interfaces look like if you want to do sensory feedback after amputation? And last not least, I dive a little bit into blood pressure control. Can we use electrical current instead of pharmaceutical pills to lower the blood pressure? And which side effects do we have to expect there? All right. So first of all, is it worth spending money into those implants? And my favorite study comes from 2014 and reflects the cost of brain disorders in Europe in 2010. And if you take those numbers, and you will find other statistics that calculate the numbers differently, but we are in the range of cardiovascular disorders and cancer together with the cost of brain disorders per year. And this is probably a good starting point. Say, well, it might be worth to look more in detail into those novel technologies to interface with the nervous system to treat some diseases. So potential applications, what might they be? We know deep brain stimulation. We know cochlear implants as the success story of neural implants over the last, well, don't know, 35, 40 years. There's spinal cord stimulation with pedal electrodes. We have had 1 or 2 decades of retinal vision prosthesis, and unfortunately, they did not work that well, and they are now bounced back to fundamental research to find out how proper stimulation paradigms should be to make the blind see really in detail. And we have electrode arrays to put on the brain or to stick into the brain. And I think most of us have seen more or less the presentation of Elon Musk and Neuralink with Gertrude, the pig, and all those things that can be stuck into the brain. So these are potential applications. And the question is, what fundamentals do we have from the engineering side? What is our task? What is my task as engineer to make something work? And if we look on the success stories, well, more or less success stories, for clinical applications, we immediately see that those implants look very robust. They have to be reliable to work over years and decades. That means long-term stability is key. And to convince your notified body and your legal authority, most companies use established material with a long material history, like silicone rubber, platinum iridium foils, stainless steel wires, something like this. And of course, you have to make your surgeons happy. So those things should be easy to implant, that you do not destroy the whole thing during the implantation procedure. When we look on the limitations, we find, well, it's not really small. The number of channels is limited. cochlear implants, as high channel implants have 21 channel max. That's not very much. If we look in one of the latest funding programs in the US, where 1 million of contacts, wasn't it something like this, expected to happen soon? And the materials are limited if we stick to those with a certain history. That means we come out with some limitations and needs. And to be honest, the needs that we have are not very sexy. So it's about cables and connectors. But that's not a topic where you can really get new PhD students to get excited on the first day. It's about packaging, and it's about the electrodes as such. And if we think about Western societies and the lifetime expectancy of a newborn, this is roughly 83, 84 years in girls. And as engineers, I cannot calculate that good. I would say that's 100 years. So implants should survive 100 years. And I don't know any electronic gadget that is so old, or where I would say, well, this could really live for 100 years. Things die earlier, and we need methods and models to predict how they work when they're miniaturized, and we have to verify and validate all the methods, and this is really pain in the neck. So before talking too much about theory, I want to walk you through some things on examples. And when I started in my PhD thesis, because my boss told me, here's a clean room, do something in it, and why not doing electrodes? I have a material for you, just start. I was thrown into polyimide, not physically, but into that topic. And nowadays, I'm really convinced that this is a good material. If you work in a clean room, you take those wafers, so slices of silicon where you put everything on, and you can make it small. You can make many things in parallel. That's the appealing thing. And we have thin sheets where we sandwich metal in between layers of plastic that should insulate, deliver mechanical stability, and electrically insulate one line from the other. Electrodes should not corrode. They should have good conductivity, and they should selectively stimulate without releasing material that is toxic to the target nerve cells. Beyond obvious challenges, the assembly of those systems, the longevity, and the medical device approval are still not on the easy side. And I will go through some details with respect to that. First of all, the stability of thin films. Well, a thin film is very thin, as the name says. And if you have had a metal sheet and a precision mechanics electrode in your hand, you will see it just is there. A thin film is so thin, if you would blow on that and it's not sticking to the substrate, it would just fly away. And that is the challenge here on those slides. On this slide, if you have a really new, pristine thin film, it looks very nice. If you stimulate for about 60, 70,000 pulses, doing nothing, that it really sticks to the underlying ground. Some electrochemistry takes place, and you have a kind of popcorn effect, and you get those bubbles, and if you touch them, they'll break, and your electrode is gone. And this has been, over the years, one of the largest challenges to make that very thin layer stick to the underlying substrate, that it does not get destroyed. And this is something that you do not see in your first round of acute experiments where you get excited, you know, putting that for an hour in a rat is awesome, but doesn't show you the problems. The problems show up after 6 months, 8 months, 10 months, or something like that. What can you do? Well, normally you have more or less rough surface and you put things together, and they might somehow stick together because of that mechanical interlocking or some physisorptive processes. But to make it really stable, you need a strong bond with a lot of energy that it sticks together. And that was our challenge. And we stole the idea from Stuart Cogan and modified it a little bit to use silicon carbide to provide chemical bonds between the metal, which is platinum in our case, and the polyimide, so that the carbon side anchors to the backbone of the organic side, and the silicon with platinum silicides anchors to the metal layer. This is the idea, and I show you some examples, or one example, how this looks like if it really sees a human being over 6 months. First of all, we had to show that it sticks. And to cut a long story short, we included that adhesion layer. The yellow stuff is our sandwich, 2 layers of polyimide that insulates the metal. The gray one is the metal, and the dashed dark gray black one is the iridium oxides to make the electrodes more stable and allow more current to flow over that. And we put that into red. Without adhesion layer, we see that beautiful dome. Looks nice, but that means the electrode is gone. Just a little bit of mechanical pressure or force, and everything will fly away, while everything sticks well with adhesion layer. The dark bluish color comes from the iridium oxide, which has a photochromic effect, and therefore it's nothing else but that effect during taking the pictures here. If you would ask somebody from outside the field, say, hey, what do you think a problem is? Nobody would come to the idea that the assembly of the electrode is one of the really hard things. And the more electrodes you get, the harder it gets. And the reason for that is that you need a lot of interconnect cables. One electrode, one cable goes to one pin of one connector. And this array here of 252 contacts has seen a non-human primate for about 17 months, and it worked well. And at the end of those 17 months, less than 8% of the channels dropped out, 20 out of 252, and they were not really gone, but their signal looked like the signal on the neighboring pin. And then we thought, well, where does it come from? And we traced that back to the connector array and found out that this is a complete line here on one pin that has gone. And the reason for that is, you might know those Omnetics connectors. They are really tight. And if you have to put your cable on and off every day or every second day, you have to wiggle it on. And by wiggling around, the forces will be transferred to your solder points and to the underlying epoxy. And then the epoxy gets a little bit loose, the water creeps in, the water condenses, a little bit salt is drawn by diffusion through that, and then you just have short circuits and see the same signal on a complete row of your connector. And the more channels you have, the more, the higher the probability that you see those things. And what material could you use? And all materials I know were not the best ones. And then we came to the idea, why not putting silicone rubber in between? It's flexible on one hand, so if you wiggle, it can be just a little bit pressed or pulled, and it's not that permeable to ions. So that means even if we have water there, it's more or less deionized water, and should have a good insulation resistance. And this is here slide for the freaks of micromachining who might be here. For all the others, they go to the next slide, which is easier. We have here adjacent contacts, and we pre-manufactured here a silicone rubber mat with holes where the contact pads are, and then we somehow glued by plasma activation this insulation sheet to both, to the substrate, and glued our contact array on top and molded everything in silicone rubber. And I have to admit, we had to do some tricks that the polyimide likes to stick to the silicone rubber. This is a quite complex process technology, but we made it work. And then we measured adjacent contact pins here with a distance of, I have to lie something a little bit below 1 millimeter. And after 22 weeks at 60 degrees C in saline solution, we still got about 20 mech ohm at 1 kilohertz insulation resistance. That means this could be an option, combining old school silicone rubber technology with micromachining to approach those challenges of assembly. So, but how does that work if we apply it somewhere? This is really the engineering side. You have to build it up, you have to assemble it, you have to make cable and connectors. That's what a lot of persons take for granted if they approach an engineer. I just want to have an electrode that works. And how does it work on the interface with the target tissue, with the brain, with a peripheral nerve, or even with nerves in the autonomic system? And I have two blocks of applications now. The first one is brain probes. So those flexible arrays being interfaced with the brain. And I start with electrocorticography arrays. That means arrays that you put on top of the brain. And we did that for basic neuroscience together with colleague Pascal Fries at the Max Planck Institute in Frankfurt. That was the first version here of a 252 channel array that was put on a macaque on a macaque brain. It worked for more than 2 years, recording only. And we've seen that we got a quite good signal to noise ratio even after 6 months, which is here on the very right-hand side, having platinum electrodes, only substrate thickness of 10 micrometers, and relatively large electrodes on that. Assembly was the hardest part, to be honest, to solder and glue all those connectors to the array. That was very promising. And we got approached by other colleagues from Hamburg and made it smaller, and they went into ferrets, where not so many groups worldwide are working with. And the arrays were also stable there. Ferret has a smaller brain than a macaque monkey. That means the curvature of the brain gets larger. And even there, those arrays with a thickness of 10 microns, and here electrodes with a diameter of 250 micrometers worked well. And the exciting thing for me was that due to the good conformability and adherence of the array to the surface of the brain, my colleagues were able to record spike-like activity over those arrays, not only local field potentials. And that means you do not necessarily need amplification circuitry, electronic amplifiers directly integrated in the array, but if it really sticks well to the surface, you're able, while switching the bandwidth during recording, to get both either local field potentials or spike-like activity that corresponds well here to multi-unit activity seen with intracortical probes. The blue shape, the blue spikes is intracortical, the green ones are recorded over this array. We played around a little bit with structures like finger arrays and with glassy carbon as material to investigate if we can use this, not here, but in other investigations to detect, for example, electroactive substances like dopamine, and to see if we can reduce imaging artifacts during MR imaging, having carbon instead of metal as electrode and track material. But I had to cut this out due to time restrictions here of that talk. Otherwise, I would have ended up with 150 slides or something like that. What we've seen is that glassy carbon GC here is not that bad, but as expected, if you lower the electrode diameter, the noise gets higher and the signal to noise is not that good. However, if you coat these electrode surfaces with a polymer, P.PSS, one of those famous conductive polymers in neural implants, you get, since this works more on the volume of the coating instead of the area of the cross-section, you get a good signal to noise, a low noise floor over all electrode diameters being here on the surface of the brain. And these recordings are taken from a rat. Years after I started with the design of the arrays, we thought, well, probably our thickness has a certain secret, because a lot of other groups reported that polyimide heavily damages target tissue in the spinal cord or on the brain. And we thought, well, probably the secret might be in the thickness and the curvature. If you have a certain material and you want to bend it, the smaller the curvature gets, the more force you have to apply. And depending on the thickness of the material, it's not only the Young's modulus, that means the flexibility, but it's also the thickness of the material in combination, which interacts with the brain. And if you have a very thin material here and you put it to the brain, during implantation, adhesion force, just by that capillary forces, just bends it on the brain without anything. If you get thicker, 10 microns instead of 4, you have a small interaction. The thicker you get, the more pressure you apply on the brain when trying to make it conformable. This was something that we tried out and found out that we were lucky, because incidentally, we had always the right thickness of our material, that we stay in that really good range, that we do not apply any force or any significant force on the surface of the brain. How can we use this knowledge now for intracortical probes? First of all, it's a nightmare. If you get really thin and flexible, how should you push a probe into the brain? It's like pushing on a rope. That doesn't work. You have to make some additional tools. And we started with that idea, can't we make things so flexible that we can bring them somehow in the brain and have those floating wires that minimize tethering forces on those probes and force interactions on the brain. We did that again with our technology, and we had some crazy but a bit stupid ideas of a horseshoe that we could use to put a guide wire in and then push the horseshoe with a small flexible probe inside the brain. And it turned out, as you can see here, that horseshoe is so large that it will destroy everything, and you don't get it through the dura and it will not work. So we had to play some tricks to get at least some signals here. It took us some years, and then we thought, well, we don't need those horseshoes. We make things very thin and flexible. Just make a hole on the tip, use a very thin tungsten wire, put the tungsten wire through the hole, push it in, and pull the tungsten wire out. And that worked well. Only thing was that I lost all the guys who worked on that, all the guys and girls, and I now have to assemble all this data and publish them by myself. So keep your eyes open, but it will take some more years on that. Those transdural shafts seem to be promising, but the question is how thick or how wide, how large should they be to result in a foreign body reaction that is minimum. And when I ask that question, sometimes there were some rumors around, you know, those urban myths where people say, well, probably 30 microns is a good size. If you are below 30 microns, you will not see so much. And when you ask, well, why that? Why not 25 or 50 or whatever? It turned out that people say, well, I think, you know, if nerve cells hold themselves by their hand or they want to see their neighbors, so might be something about diffusion length of certain agents, and it might be about the width and the thickness and thereby the moment of inertia. And we tried that out, and I hope you can see the figures here, the pictures as good as I can on my screen. The left column is 100 microns wide, the right column is 30 microns wide. And well, the wider it is, the wider the glial scar that's shown here after 4 weeks. That's obvious for us completely surprising, and these are preliminary data, and we're currently reproducing them and are on the way and publishing them with more animals. After 16 weeks, you do not see much or nothing with the small devices, while you see a massive hole here, nerve damage, retraction of neurons, whatever damage on astrocytes and glial cells here on the other side with a larger arrays. And then that is a good indication that you should be really as small as possible. And this is now reproduced by many other groups which use a lot of fancy technology. And that was a kind of competition for us to ask ourselves, can we do that with our simple technology? And we did that and made probes up to many, many layers that are comparable now. And that was funny because we had them. We were a little bit slow in getting figures and publishing them. And that was coincidental with the first announcement of Elon Musk. So we had incidentally nearly the same size. And he came up with that not very usual display of the electrode impedance, and we recalculated our data. So we have been comparable with that. And in collaboration with Juri Buzaki's lab, where we could show that these probes are able to record single unit activity in rodents for at least 6 months. Still work that is not fully published. We have the first results here. Publication is underway, but that shows that you can use standard technology that has been already established in many preclinical trials to go also towards higher integration densities. So that was all preclinical, so fundamental neurosciences. And my dream from the very beginning was to develop something that goes into humans, right? I don't do that because I want to make technology smaller and smaller. I use that technology because I want to make something useful for applications in humans. And might have been around 2008, 2009 that Silvestro Michera from Pisa and now from Lausanne EPFL approached me, and we assembled the first or second grant proposal about sensory feedback in artificial limbs. And over the years now, we had 4 patients with upper limb amputation, 1 30 days patient, the first in human trial, and 3 patients up to 6 months. And then another 3 patients up to 4 months in Belgrade with lower limb amputation. And I'm pretty sure Silvestro showed you all the videos during his talk. So I want to focus mainly on the electrode side of that. What does that mean if you develop an electrode? Which challenges do you have? And what are the results that I'm very proud of in that study? So we were all proud of the whole outcome. And for those of you who haven't seen Silvestro's talk, the idea was to have myocontrolled hand prosthesis with electrodes implanted in the nerve to give sensory feedback through a sensorized prosthesis, and the person is blindfolded, that he or she does not see what kind of object she or he should grasp. The time electrode, time stands for transversal intravesicular multi-channel electrodes, a really long, difficult word, is micromachined of that polyimide technology. That's here the front part in this picture from the whole electrode. That's the part that you cannot see due to that surgical thread here and the needle, which was then assembled to a helically wound cable here and a connector. And this is the only video that I've brought with me here. It's about the concept of that time electrode gets implanted above the elbow. The electrodes have iridium oxide coating in polyimide embedded. This is not the thickness, but the diameter of the electrodes, 80 microns. They can deliver 120 nanocoulombs of charge. And we have 16 active sites, 7 of them looking to the left-hand side, 7 to the right-hand side, and 2 counter electrodes to close the electrical circuit. Surgeon implants them by pulling them with needle and thread through the nerves. These are the yellow bodies here. And the cables are then routed with some strain relief through the skin. We have 2 electrodes in the medium nerve and 2 in the ulnar nerve. Prosthesis is sensorized with pressure sensors. The data are processed in a computer and then fed back as stimulation pulses. And if you select the right stimulation pulses and the right electrodes, the person can discover the object, shape, and the compliance. That means the stiffness of the object. This is a nice animation. The question is, how does that work in real? That's the first prototype of the artificial hand that has been designed in Italy. You might hear that this is very, very loud, and that is the reason why this person got music on the ears. So, and our first patient was what we call him the plug and play patient. That was amazing for us. He was able, after 2 days, to intuitively understand the signals and use it. We have had other patients that needed more time to really learn that stuff, but after, let's say, 4 weeks at latest, they were able to intuitively use those prostheses. Encoding is the key for success. If you have just fixed frequency, fixed amplitude pulses, you can get something. But if you play around with changing pulse to pulse distances and adapting the pulse shape over the stimulation train, then you get also telescoping and better embodiment. And that means that people feel their hand moving from the stump site, where they feel it before getting that prosthesis, to the original site where it has been before amputation, and they feel the limb really as part of their own and can increase manual accuracy and sensitivity and stuff like that. Afterwards, I had the chance to jump with those implants also in the lower limb world. And we were able to deliver electrodes for 3 patients there who had a very nice distribution of perceptions over the foot sole, as well as over the ankle and the lower limb, to get the impression about knee flexion. They have been amputated above the knee, and so it was important to give them also some information about the knee position. I skipped the slides of those. What I want to show you here is that the different colors code for different implants. So we had again 4 implants in branches of the sciatic nerve. And 1 electrode means this is 1 implant with up to 14 contact sites. And here, if we stay with red, we had here in patient 1, S1, 6 sites that were able to elicit meaningful sensation in that patient. In the other patient, we had 13. In the third patient, we had about 12. And that depends how the surgeon in detail implants the electrode, and you have to check it out channel by channel afterwards, what the patient experiences. Is this vibration, pulsation? Does it feel like touch, or is it pain? And if it's pain, you should not use that electrode contact, of course, at least not for detecting touch or vibration. You probably might use this as a kind of alert system, if you would imagine that you could include a temperature sensor or something like this in a prosthesis if something wild is happening, that you can use that channel as immediate alert, but this is not yet planned. All in all, that was sufficient. So was sufficient to deliver perception there. And that's probably the good news, that you can work with a relatively low number of active channels at the very end to deliver feedback. So it's not necessary to have hundreds or thousands. In our case, is 4 to 5 channels maximum were then used to deliver the feedback for the performance. As you've seen, we had percutaneous wires. This is still the limiting factor, but when we started that study, we were not sure what the outcome is. And if you wait for another 5 years to get a fully implantable system, and then you find out that you chose the completely wrong parameters for the electronics inside, that doesn't help at all. And therefore, we thought it's better to investigate that first. And you can only ask a human what a human feels. We have done, of course, a lot of experiments with pigs and with rats, but you can never ask a pig about the question, how does it feel? Do you like it or not, right? And this is the point where you really have to go to humans. One thing I learned after my first visit ages ago in Case Western was that it's good to have something like a quality management system or a lab where you have control over the manufacturing boundary conditions, and this is what we did in my place. So even though some persons still hate me for that, I've set up a quality management system according to international standards where you have to document everything. And so I still can sleep very well if we do something like this, because the essential requirements, that means all the safety issues, have been covered during design development and testing then up to implantation. The question, however, always is, how good is thin film technology? And if you might ask persons from pacemaker industry, they might get mad about that. They say, well, you know, cochlear implants, cardiac pacemakers, they have, let's say, 1/10 of a millimeter of electrode thickness. And if you stimulate over 50 years, there's still 20 micrometers left. So we start with 1 micrometer. If that corrodes, there's not much what is being left afterwards, right? So we have to look on that. And we had here with our upper limb amputees 3 persons, and we checked up first in vivo impedance over time. Have been implanted here up to 25 weeks. And the right-hand side is the contact of eliciting sensation. And that is the important part, because here all those 3 lines go down. I show you on the next slide why these 2 lines with the patients went down. So we were really completely upset because we lost a lot of electrode contacts over the weeks. And of course, some competitors said, that's the thin film part. You know, we told you, that's the thin film. It's much too thin, it doesn't work. And we changed some things and received then here a performance of about 80%. And the reason why this ended here is not that everything dropped out because the experiment was terminated after 20 weeks and the subject got explanted. And after, and it's a pity, and I wanted to switch to the next slide. The reason that the electrodes dropped out was that nasty connector part here. We wrote a beautiful manual how to handle the connectors. And after 10 weeks, we got an email saying that the patient is now able to disconnect the connectors by himself. And we thought, well, we wrote in the manual, you need 2 hands. This guy has 1 prosthesis, so he has a maximum of 1 hand. And then it turned out nobody read the manual, and they were all pulling and pushing here on the cable instead of using the connector by the fingers. And by that, as you see here in the upper row, all those wires broke, or a lot of wires broke here immediately in that transition and were no longer connected. And that is the reason, going back, why we had those dropouts here. Unfortunately, patient 1 and 2 were implanted shortly after each other. That means when we got the message, was already too late to change the second implant. But we changed the implant for the third patient with a very simple thing, just adding more silicone rubber here that you cannot really destroy that part. And the outcome was much better, so increasing that. Why do I tell you that? I tell you that because I believe that a lot of failure rates come from the easy things, not the really highly sophisticated technologies, but from some other parts that might be of underestimated complexity. But the question is, what does the thin film do? Going back again here, we see at least in that red line here, the electrical performance, so eliciting sensations up to the very last day is really high at 80%. And then we got the explants, and the surgeon took care that the patient is damaged least during the explantation time, and that means the electrode is damaged most if you take care of the nerve. And we've seen things like that, where you see kinks and scratches here that tweezers, scalpels, and whatever have tortured the electrodes. And having now done comprehensive investigations with light microscopy, that's the first row here, with polarized light, that's why you see false colors. We have white light interferometry showing height differences here by false colors, and electron microscopy here showing some failures. We believe that the worst failures occur just due to handling during explantation. This is here when you want to pull something out and you, ouch, that was the surgical tweezer and it goes through the device. While a lot of electrodes looked good after that, and we did in parallel some in vitro studies where we stimulated up to 4.5 billion pulses, which is about, I have to lie, between 6 and 10 years of 8-hour stimulation a day with the usual 30-40 hertz, where the electrodes stayed stable, so that we are really convinced that we can go ahead with that technology. Question is, can we use the same technology or similar technology also in that idea of bioelectronic medicine? So stimulating the autonomous nervous system in a way that we can cure or treat diseases like hypotension, rheumatic arthritis, COPD, chronic inflammation diseases, things like that. I'm co-founder of 2 companies, as I already indicated on my title slide. And while Cortec is more delivering electrodes for others to investigate scenarios, Neuraloop focused on vagus nerve stimulation and treatment of hypertension by vagus nerve stimulation. And this is what I want to focus now on the last 5 to 10 minutes. So what is the neuromodulation of blood pressure? There's one branch of the vagus nerve that triggers the baroreflex, so the arteries dilate and the blood pressure goes down. This is here a red vagus nerve. The blue part is the standard vagus nerve. This is here other tissue around, and the yellow part is the nerve branch which carries the pressure information of here the aortic branch. If you would now stimulate the whole part of the vagus nerve, you get reduction in blood pressure, high frequencies due to the species that has been a rat, but you see that you reduce significantly the heartbeat rate. That is not good. If you want to lower blood pressure and you have a heartbeat rate then of 30 or 40, that's not very healthy. However, if you take a multi-channel cuff electrode and use those electrodes that are closest to this yellow branch, then you get a reduction in blood pressure without a significant decrease in heartbeat rate. So this is how it looks like. I mean, you are the cradle of cuff electrodes, so I think I do not have to explain you that too much in detail how a cuff electrode works. If you stimulate, for example, about that electrode and increase the current, then you approach that branch best with least interference to all the other nerves. Question is, does it work if you are under medication? Because if you are a patient or a person having 240 to 140 blood pressure and you found your medication that you can probably push it down to 160 or 150 to 90, then you will not sneak out of that medication for an implant, right? That's too dangerous. And the question is, does it work? And we did some studies here. This is the control, and this heat map shows the stimulation electrode on the y-axis, the pulse width on the x-axis, and those lines here are the repetition frequencies. And we find in the control animals without medication, large areas where you can reduce significantly the blood pressure by about 30 millimeters mercury. And having here one of those standard medications, it's more difficult. That's obvious, but you still find those islands here where you are able to reduce the blood pressure at least by 20 millimeter mercury. One question, of course, is the rat equal to the human? And we all know what does work in the rat does quite often not work in the human. We learned a lot of lessons over that, had success in pigs, had no success in sheep due to variations in anatomy, and found out that it is important to have very phase-triggered stimulation here to the heartbeat instead of having a continuous stimulation. And this is now being commercialized and all the animal experiments have been finished, and Neuraloop has submitted all the documents now to some legal authorities, and hopefully clinical trials will start in this year soon. So to conclude, what does that all mean? To conclude more on the high level, I think I could show you that there are alternatives to other treatments using those neuroprosthetics and also neural implants in bioelectronic medicine, and also in sensory feedback or in fundamental neurosciences. Well, we should keep in mind that they do not have only effects, but also side effects, and that we still have only few approved medical devices and that there is a long way to go for more of them. The distribution of all the treatments, of course, depends on the performance, on the patient acceptance, and I think also on reimbursement by health insurances in countries where there is a proper reimbursement system. At least in Europe, this is one of the killer applications if you get the approval of reimbursement by health insurance or not. And I think I could show you that engineering and medicine have to go hand in hand for success. And I strongly believe in person that those developments, especially if it comes to changing things in the brain, should be accompanied by societal discourse to really inform patients and make our minds what we should do in society to shape, to deliver better quality of life for those who need it. With that, I'm at the very end. I think I bit over 45 minutes. Thank you very much for your attention, and I'm happy to take questions.
Well, thank you very much, Thomas. That was fantastic. We're open for questions. You can either just ask out loud or send something to the chat. and I'll relay it. And while people are getting their questions together, it's kind of not a personal question, but you mentioned Elon Musk a couple times. I'm just curious what you think about his approach.
Don't know the English proverb for that. Germany would say there are two hearts beating in my breast. So I'm very, very impressed about the technology development that he showed up in few years. So by really having enough money and concentrating or pushing a whole team towards a common goal, for me, it's impressive how fast that technology development proceeded and how he succeeded. On the other hand, thank you, Ron. Yeah, I do not get mixed emotions. On the other hand, this way of presenting those things and mind reading and controlling your mobile and all of that stuff that you might have seen is something which I do not like at all, because I believe it raises the wrong expectations in persons and patients who really are desperately looking for help. And I do not believe that these devices will deliver the help or the performance that is needed for medical devices in spinal cord injured patients, for example, in patients with severe neurological disorders or something like that.
Yeah, I agree with you completely. I mean, our emphasis is on rehabilitation, and most of their at least disclosed applications are not. So it's a little bit of a mismatch there. I wanted to ask you about the ECOG recordings that you showed spikes, and do those happen immediately? I mean, when you put them when you apply the arrays in surgery, do you get the spikes immediately, or does it take some time?
Good question. I believe these data were from chronic recordings. The diameter of the electrodes with 250 microns is still, let's say, large enough. That means you do not trap air bubbles or whatever underneath. So therefore, the signal quality is relatively good from the beginning on. But we discovered in smaller electrodes, when they're around 20, 30, 40 microns in diameter, that it might take some days before whatever is there is resorbed and you have a good contact. So, and sometimes it takes up to that magic 10, 12 days where you have the first acute inflammation period, and after this settles down, the quality of recording stabilizes.
Yeah, it would be very nice to be able to have some in the brain interfacing work we're doing. You need to implant the electrodes and just wait, right?
Yeah,
and
yeah.
it would be very nice to know some things. Are you in the right place? Are the electrodes not broken? We do some testing, but yeah, it's always an issue. Or a concern, I would say. Okay. Other questions, anyone?
Yeah. May I ask a question here?
Absolutely.
I was interested in the thin film, the changes in the structure, that it looked like to be able to build that bubble in there, there would have had to been an increase in surface area for that to have happened. And
Yeah,
suggesting is, I guess where I'm wondering is there some possibility that the stimulation was resulting in a crystal structure change that would allow that expansion in there? Because that kind of thing certainly does happen with iridium oxide. But you said this was platinum.
No, that was iridium oxide.
Oh, okay.
We did it on platinum.
Yeah, it's go ahead.
And the change in the crystal structure seemed to happen in the platinum, not in the iridium oxide. So the iridium oxide, I have not shown you the focused ion beam electron microscopy pictures here. So the iridium oxide looks pretty stable. What we have seen is that we experience changes in the platinum thin film, where grain structures are, a single grain is at the thickness of the whole thin film structure. And then you get nasty stuff with oxygen and hydrogen at the interfaces of the grains, and that enlarges the structure. I doubt a little bit that you can use it for something good. We have experienced that the contact between the electrodes and the nervous tissue is relatively good. So since everything is so thin, plus having sharp edges, I have experienced that as in early experiments in cuff electrodes, most of the tissue grew around and did not want to grow in. So you do not have much tissue in between the electrode contacts, foreign body tissue between the electrode contacts and the target tissue. I once tried it out to make those domes on purpose, and found out that the tissue reaction was larger than having just the flat surface. That could have been incidental, but so far I'm quite happy with my flat electrodes.
Well, thank you.
Other questions? Well, if not, I'd like to thank you.
Oh,
wait a
here
minute.
comes one.
Yep, we have one. I'll let you read it, Thomas, I think.
Yeah. So impressive specificity of sensations related to the knee and the foot elicited from implant locations above the knee. The human test bed was for 3 or 4 months prior to explantation. Are there plans for longer term trials, and what are the issues involved in chronic use that might drive changes in electrode design? Probably the first part, in Europe, we have some problems with legal authority and ethical boards, if we already have those results, to argue why we should go longer than 3 to 4 months, since the percutaneous cables always come with that well-known risk of infections. So this is still under discussion in the larger group, if we should go for a selected smaller group of patients, let's say up to 12 months, to look in that. What might drive changes in electrode design? Yeah, good question. I think if we would know better how many contacts we really need, what an optimum size is. So, you know, the larger the contacts get, the more current you can pass through, or term it the other way around, the lower the current density is, so they might last longer, but the selectivity goes down. If you have smaller contacts, you have more, that means more cables. Difficult to say. So we are still playing around there. And one of the major things that we have to consider is the question, how do we get this thing towards a fully implantable system? We do not know which contacts we can use a priori. That changes from person to person, as you have seen. So we need 30, 40 contacts to select 3 to 5 or whatever, how many for the chronic use. But having 40 wires and connectors with 40 contacts, there's nothing on the market that's long-term implantable. And this is the challenge, how to approach that, to co-develop connectors and cables with that, and the electrode electronics. So it's currently less the electrode design, but more the system design to get that nice selectivity in an implantable system, because you cannot resolder or rewire things after some weeks. I mean, it would be ideal to have percutaneous wires to find out these 3 bodies are the best ones, and just connect those 3 wires to an implant and do it in 2 times. But I think that doesn't work, and this is currently our state of discussion. So how to combine that, how to make that really chronic, how to get it fully implantable.
Okay, last chance. Okay, well, Thomas, again, thank you so much for joining us late in the evening for you. And that was a fantastic talk. And yeah, I hope to see you in person sometime soon.
Yeah, likewise. Thank you so much to be all here in my talk. Was a real pleasure to me, and hope to see you soon in person this year. Probably not, but probably the next one then. Thank you so much.
All right, bye. See you, Thomas.
Bye-bye.
Thanks, everyone.